since I only have 15 minutes, I'm going to go ahead and jump into my presentation to make sure I try to stay on time here. I think Craig did a good job of just talking about the fact that there's no single unified definition for NAMs. But I do want to point out, as he alluded to as well, that there are, within these different definitions, there are some commonalities that are generally seen. For example, NAMs are, in these definitions, NAMs are mentioned to improve human relevance and also being able to reduce the reliance on animal studies. So I just want to point that out just to introduce NAMs and not to be too repetitive for what Craig said. Some of the potential benefits of NAMs for regulatory decision making, I want to go through this, to improve the modeling of human toxicity compared to traditional animal studies. And as a result, you get increased accuracy and relevance of human health assessments for chemicals. Also, these NAMs are purported have the potential to provide more detailed mechanistic information on toxicological effects of chemicals. And this is important because that can get you down to the details of the hows and the whys of how a chemical is exerting its potential effects, which is something that the apical endpoints that are seen are tested in traditional animal studies don't give you that level of information. NAMs have the potential to reduce reliance on animal testing, which ultimately can lead to reduced costs and experimental costs as it relates to evaluating these chemicals. And if you take all the 4 points that I just mentioned ahead of this one, you can see that NAMs have the potential to accelerate the regulatory decisions, and as a result, could have the potential to increase or make the product approvals more efficient. And last, I want to point out that in contrast to animal studies, these NAMs have the potential to be more higher throughput, give more higher throughput results. And as a result, you can test larger numbers of chemicals more efficiently. And that's important as well because there are a lot of chemicals that do not have as much chemical data, or some of them don't have as data as all as relates to its toxicological effects. And so being able to leverage these NAMs to be able to fill some of these knowledge gaps as it relates to potential effects, it would be important and useful, especially in the regulatory landscape. So here, I just wanted to show an image here just to kind of give you an idea of how NAMs can fit into the human health assessments of chemicals. This first row at the top, it just shows your traditional approach to chemical assessments that involves the use of animal testing. I want to focus more so on that bottom row, where you have these human relevant models, NAMs, which can provide some information as relates to potentially, the dose response information on these chemicals. And as a result of having the dose response information and additional information, you may be able to bypass the use of animal testing and get to, estimates of human risk, which could lead to, being able to get the exposure levels that could be considered safe for human exposure. So just want to provide that because I know a lot of people like myself, I'm more visual. So to have an image like this to help reinforce the point. Here, as we all are aware, I think most of us are aware that our modern chemical assessment toolbox is expanding, and it's expanding at a rapid pace. I don't intend to go through any of these in particular detail, but just show you just the amount of tools that are available to us, the non-animal tools that are available to us that can help assist us as we're making regulatory decisions. There are many NAMs that have been developed. I know in industry, NAMs are being developed in academia and other areas. However, there are only a limited number of NAMs that have been accepted or adopted for regulatory use. And I just want to go through some examples of some of the challenges. I know Craig just mentioned a few of them. This is not an exhaustive list here. This is just some of the ones that I wanted to highlight. The validation process is a labor-intensive, time-consuming process. That's one of the big issues. Next is, although our mindsets and thought processes are starting to shift, I think there's still that pull to rely more so on the traditional animal testing as the gold standard. And to piggyback off of that point, the performance of NAMs in general are being benchmarked against this animal testing. And as most of us know, animals have limitations in their ability to predict the human biology. Okay. And another point that I want to bring up as far as a challenge is related to being able to evaluate and look at the data from the NAMs and understand how is it relevant to human biology. Okay, and so this brings me to a quote here that tries to I try to think about this quote to keep me in the proper perspective. It's a quote by a statistician named George Box. It says essentially all models are wrong, but some are useful. I think that's important to keep in mind, especially because throughout history we have been using a model, and as I just said, it's the animal models. They're not perfect. They have limitations in their ability to evaluate and assess human biology, and we've been traditionally looking at those over the years. So it tries to help me not to be as skeptical about NAMs in certain aspects, because we've been, dealing with and using an imperfect model. NAMs aren't perfect. I think they have potential usefulness, and we have to just identify where that information is most useful and where we can leverage that information. So just keeping that in mind. No model is perfect. Other challenges, there's a limited ability of NAMs to cover complex regulatory endpoints. And I want to put more context around that. I want to say that they're individual NAMs. A single NAM by itself is not going to be a one-to-one replacement for, potentially say more so like a whole animal study or something like that, even though I just kind of downplayed animal studies. But I will say that NAMs individually, one NAM is not going to be able to cover complex regulatory endpoints such as systemic toxicity, development and reproductive toxicity. Okay. Also, another challenge is the ability of NAMs to model chronic exposure scenarios. I think there's a lot of work that needs to be done in that area, and that's relevant here in this space here. We're talking about foods. Food consumption is a chronic exposure scenario, and I think NAMs, we have to figure out how we can leverage the NAMs to get that level of information. I'm going to skip that last point for the sake of time. Another challenge, and I just want to bring this into context from a regulatory perspective as well, is that one size doesn't fit all, and I'm just going to use EPA and FDA as examples here. EPA and FDA are made up of multiple different programs, offices, and centers, and they have unique needs, decision frameworks, and regulatory considerations. And if you look to the far right, you'll see that there are multiple different types of products and chemistries that each of these program offices and centers are dealing with. And as a result, you also have different exposure scenarios. So at the end of the day, you can imagine it's unlikely that one particular NAM or an approach is going to be able to be sufficient to address all of these different types of needs, all these different types of scenarios and chemistries that we're dealing with in a regulatory standpoint. So since this is the topic here today is foods, I just want to point out, I think most of us are aware of this, so I'm not going to spend much time at all on this. The potential chemical hazards in foods. We have food additives, we have processing contaminants, food packaging. We got migrants that come from the food packaging that can get into the food. We have environmental chemical contaminants, heavy metals that can get incorporated into food through the soil, air, and water. Pesticides through agricultural use, as well as natural toxins such as mycotoxins and alkaloids, are for an example. So we're thinking about developing NAMs for food chemical safety assessments. I just want to point out some things just to keep in mind. And just from the standpoint of when you try to compare this to the pharmaceutical and drug space, food chemicals are not designed to be or intended to be biologically active at levels that are typically consumed in the diet by the general population. So any effects at the typical dietary levels are expected to be minimal or absent. The exposure scenario, as I mentioned, is you got the low dose chronic exposure via the diet, lifetime exposure. And so these NAMs that you're developing, you have to take that into consideration. Is the NAM going to be sensitive enough to be able to capture some of those low dose and assess the chronic exposure scenario? Unlike pharmaceuticals as well, there's no risk-benefit assessment that was mentioned a couple of times earlier in the first session. Food consumption shouldn't be any risk. Any food should be safe in general, and so that aligns with the principle of reasonable certainty, no harm. And the last point I want to make here to think about, I think Christy mentioned this during her presentation, is you have different exposure across broad populations, including you have pregnant women, elderly, you have infants, just to name a few. So these are some of the things just to keep in mind as NAMs are being developed, specifically in the context to evaluate food chemicals. Validation. Validation is the in tradition has been the cornerstone for broad regulatory acceptance of the test methods. But as I mentioned previously, there are challenges with validation, and that's where qualification comes in. It allows for a case-by-case acceptance of tests to support regulatory use, even if it's not fully validated. And that provides more flexible and practical approach to evaluate the suitability of NAMs for a specific context of use. And I think this is an important point here. With this, I think with the changing and the currently consistently evolving landscape of NAMs, I think qualification is more suitable because it allows for that flexibility that a validation process, is being, more of a time-consuming, labor-intensive process. I think qualification is going to be one of these things that's going to be more useful to kind of push for the acceptance and use of NAMs. So objectives of NAMs, qualification process is to facilitate more efficient regulatory use of suitable NAMs for human health assessments based on expert panel review to ensure that they are scientifically valid and fit for purpose. And this allows a NAM to be used in a specific context of use without the need to evaluate its suitability for each subsequent human health assessment. So quickly, I want to go through just talk about context of use really fast. I know Susie's going to talk about this next, so I'm not going to go into a lot of detail. But context of use describes how a method is intended to be used and its regulatory purpose. It involves a couple of components. You have your regulatory application, which describes the specific purpose of the NAM, whether it be hazard identification, screening, or prioritization, or even being used in a risk assessment. Okay. You have scope and boundaries, which is the type of chemicals, the exposure scenarios, and the endpoints that a NAM will assess, as well as its limitations. I think that's important. You do have to be knowledgeable and understanding of the limitations, not only just the strengths. Okay, so limitations are just as important as the strengths. The technical performance is the next component, and it describes the level of reliability, reproducibility, and accuracy needed for regulatory acceptance. And last, we have the regulatory framework, which ensures that a NAM aligns with the existing regulatory guidelines and risk assessment paradigms. I'm going to skip this one because I only have 5 minutes, and I know Susie's going to talk more in detail about, a qualification process. So I'm not going to steal her thunder here, and just going to the next slide. Here, I want to just provide a couple of examples of NAMs that have been established for regulatory use, or those that are progressing in that direction. Skin sensitization testing, I think that's one of the more notable, more popular, or well-known test that has gotten NAMs that has received regulatory acceptance. It's a defined approach for skin sensitization hazard identification. It's based on a battery of in chemical and in vitro tests. For the sake of time, I just list here. You see the NAMs, the different NAMs that make up this test. It is recognized as a full replacement for the animal-based local lymph node assay, and it's accepted by a number of regulatory agencies, including FDA and Health Canada. The next one is the comprehensive in vitro proarrhythmia assay. It is a battery of in vitro and in silico approaches to assess the proarrhythmia potential of drug candidates. Again, you see the list of the different NAMs that comprise this assay here. It is designed to supplement or replace animal testing for drug-induced QT interval prolongation, and it too has been recognized by a number of regulatory agencies, including FDA and Health Canada. The last one is developmental neurotoxicity in vitro test battery, and as the name says, it's a battery again of neuronal cell-based assays to screen and prioritize chemicals for potential developmental neurotoxicity. This assay here is currently in progress. For example, not for example, but there is an OECD recommendation document that discuss these assays, and it's anticipated to supplement or replace animal testing for DNT. And this is a set of assays that EPA has been heavily involved in, as well as some of its European partners. And it's a set of assays in which the FDA Human Foods Program in particular is interested in, as far as being able to evaluate the potential DNT of food-related chemicals. I want to talk about two organizations or groups really briefly. Not these are the only ones that are pushing forward to advance the use of NAMs. in regulatory decision-making. The first one is the International Liaison Group on Methods for Risk Assessment of Chemicals and Food. It's a mouthful, but in short, we call it ILMARAC. It is established through a partnership with FDA and EFSA. It's a network of over 30 governmental and intergovernmental organizations that includes food regulators from around the world. The goal is to support the development of new risk assessment methods for food-related chemicals. So this group focuses primarily on foods. It promotes global harmonization of risk assessment methods and creates synergies between participating organizations. Now, ILMARAC developed a NAMS working group, which is tasked with identifying priority actions to increase the use of NAMS in food safety assessments, to identify key data gaps that may be addressed via NAMS, and to promote the application of NAMS in risk assessment practices. The other group that I want to highlight is called Complement Animal Research and Experimentation Program, or Complementary. It is a major initiative launched by the NIH in 2023 to accelerate the development, validation, and adoption of NAMS to complement or replace traditional animal testing. There are three components: Technology Development Centers, Validation and Qualification Network, and the NAMS Data Hub and Coordination Center. I just want to focus in on the last one here, is the Data Hub. It is a central hub for data integration, transparency, and coordination across NAMS initiatives. And it is intended to ensure methods, results, and best practices are shared across scientific and regulatory communities. And the reason why I want to point that out is because I think that's an area in which, there is a need. There are a lot of activities going on in NAMS, and I think having like a repository like this where, NAMS developers and scientists can go to a place and kind of see what's going on, what NAMS are being developed, what stage in the process they're being developed. I think that's an important thing to highlight as far as a need, and I think this program will be addressing that. And in addition, this hub will integrate information and knowledge from those other two components. So I'm going to finish up here with the roles of the regulator in advancing development and use of NAMS. So first, I want to say that the regulators should not just be considered just the end user. I think they have a very important role. We have an important role to engage with developers early in the process. And I think by engaging early in the process, we can ensure that the I saw the time and I just got thrown off. That the regulatory relevance is incorporated into the process earlier on in the process, as early as possible. Regulators should identify knowledge gaps to help guide toxicology research and address regulatory questions. We should also be involved to make sure that we're developing clear and transparent guidance needed to qualify NAMS for regulatory acceptance. I think this is very important because that's going to help potentially just get us faster, more efficiently from that innovation step to acceptance, if we're able to, as regulators, provide that clear guidance and transparency, on what's needed for acceptance of these NAMS. And last, which is also very important, is to invest in training, not after the fact, but as these processes are going on. Again, early in the process, invest in training programs, capacity building to increase regulatory review the regulatory reviewers' familiarity with NAMS. Understand the strengths and limitations of these methods, and also how to interpret the results in a context, within the context of these regulatory frameworks, which we're utilizing. I'm going to skip this slide here. I'm just going to finish up and say, alone we can do so little. Together we can do so much. This is a quote by Helen Keller. I like that quote. And change takes time, but if we all work together, it can happen. So thank you.